Hi everyone. So we're going to start our new unit today on Mendelian Genetics. I know that this is not an ideal way for you guys to learn this material, but I appreciate that you guys are just working with what I can give you right now. Please feel free to ask me questions through email. I'll do my best to answer them. And I don't know how long I'm going to be gone for. I don't know when I'll be back, but I'm just going to do my best to get back to you guys as soon as I can, okay? So we're going to start off with Mendelian Genetics. I'm just going to talk about the history of Mendelian Genetics a little bit, give you some vocab. Some of this you guys might recall from junior high. I know you guys make jokes about grade 9 science, but this is where you would have learned it if you did. So I'm just going to go through some of that vocab. We're going to talk about monohybrid crosses, which are very, very straightforward. You guys will catch on to that super, super easy. I'll have you guys do a little bit of practicing. I'll give you the answers so that you know that you're on the right track, and then we'll go from there, okay? So I think what I'm going to do is, instead of having you guys write down the notes as I go through them on the board, I'm just going to write down some notes here on my iPad, and you guys can kind of follow along with the version that I've written. I'll have somebody print that out and give that to you, so you can kind of note some of the most important things from that. I'll also just give you guys a bunch of YouTube videos that'll help kind of reinforce some of the things that I'm talking about. But again, please don't hesitate to ask me any questions on email. I will answer them as best as I can. So today we're going to start off with just some Mendelian Genetics vocab. So first off, when I say Mendelian Genetics, it's referring to the work of an Austrian monk named Gregor Mendel. You will want to know his name. I saw a numeric response question specifically come up about different scientists in genetics last year and what their roles were. So Gregor Mendel studied inheritance patterns, meaning genetics, in pea plants. And what's super cool about his story, I'll show you guys a YouTube video, just some of the history, but he lived in a monastery, and monasteries, monks, not normally associated with science, but he was passionate and noticed some different patterns and just really cool how he studied. Now, the pea plants are significant for a few different reasons. Number one, they had many characteristics that were either or. What I mean by either or is you'll see a lot of examples about pea plants being tall or short, wrinkled or round, green or yellow, axial or terminal buds. Like, there's so many different examples of traits that are either or. This is significant because they're very simple patterns to observe, and so it's really easy to trace them and to do those monohybrid crosses, we call them, that you guys are going to learn about today. So pretty straightforward. The next reason why pea plants were important was because we could self or cross-pollinate. What that means is that they could reproduce asexually, so that's asexual, or cross, which would be sexual. This is important because asexual reproduction, you know that we're not having any variation, we're not having any other genes coming in, so the offspring should be identical, so the patterns should be observed, very straightforward. So you have true breeding plants, we call them, through asexual reproduction. There's no other source of genetic information to change that. Versus cross-pollination, where you could take two plants, cross them together, and then observe the patterns, the frequencies, all the different things about the offspring. So if you had a green and a yellow, how many offspring came off green? How many came off yellow? So that was really significant. We could actually be quite certain where the genetics were coming from. And so they were really easy to study in that way. You'll hear me talk about self and cross-pollination throughout this unit a little bit more. The last thing that was really good about pea plants is that they could reproduce very quickly. So he could test pea plants and observe those characteristics and look at many generations in weeks versus months. In comparison, if you think about humans, where generations take decades, right? We want to be able to study them quickly so that this person can really the person studying can really understand the patterns that are happening. So those are all really, really important reasons. And this is something that you might want to highlight or star. I put it in a box inside of your notes, but just something that you'll reference. It probably will come up on some sort of assignment question, okay? So that's Gregor Mendel. There are two principles that came out of Gregor Mendel's studying. The first is the principle of dominance. Okay, so that's the first thing that's written on the left side. Oops, I spelled that wrong. Okay, so principle of dominance, which just means that some genes are going to be more dominant than others, and dominant genes are always expressed. That's going to come into play with our vocab. Dominant genes are always expressed. That's going to give us our genotype and phenotype that we're going to talk about. Now, you guys will know we're going to talk about alleles like every class for the next couple of weeks. Alleles are super, super, super important. It just means the different forms of the genes. And when we're talking today, we're going to start out pretty straightforward with like pea plants that have either or. So you have a dominant or a recessive allele, okay? We always use dominant. Dominant alleles are always shown with a capital letter. So for example, if wrinkled, I used round. If round is dominant, we give it a capital letter. If we have the other allele, the one that's not shown, the one that's recessive, so that would be, for example, wrinkled, we would give a lowercase. Make sure that you're using the same letter for both of them, okay? So if you're looking at a trait and you have the dominant and recessive, generally we always take the capital letter of whatever the dominant trait is. So you see I chose a capital R for round, okay? If wrinkled was dominant, I probably would have chose a W. Now, because this is something that you guys are going to be doing multiple choice, numeric response, it's not super important what the allele is. You really could choose anything. You could choose the same allele every single time regardless of the trait. If you wanted your capital dominant allele to always be an A and recessive to always be a lowercase a regardless of the trait, that's totally fine too. Really, except for assignments, this is the only thing that you guys are going to you're the only one that are going to see this, okay? So dominant, capital letter, recessive, lowercase letter. The thing that I would just advise for you guys when you're doing this on your own, because a lot of times you are going to be choosing the capital letters that you're using in your crosses, choose something that has a very distinct difference between the capital and the lowercase. So for example, S and S, something that's a little bit tricky because you either have to make it your big S and little s very dramatic, or you have to pay special attention. Otherwise, it's really easy to get confused. I'm sorry, you can probably hear my cat in the background. He's having a moment. Just ignore him, okay? Now, the next thing that I wanted to go over, the second principle or idea that came out of Gregor Mendel's work, is the principle of segregation. Now, this directly connects to what we just finished talking about with cell division, which basically says that traits are sorted or they're divided up independently of each other. So in pea plants, pea color, pea shape, plant size, they're all independent of each other. They have no connection on how they're distributed. So one plant could get tall, one plant could get wrinkled, the next one could be tall and round. So they're completely sorted independently of each other. So paired alleles segregate during meiosis, and they're completely independent. There will be some sort of question at some point just asking you to acknowledge that all of these are assorted independently, okay? Now keep in mind that when we talk about gametes and sexual reproduction, which is how we're combining and studying this, right? We don't generally study self-pollination or asexual reproduction because there's no variation. When we study genetics, we are studying sexual reproduction. So of course we're going to be talking about gametes. And gametes are always going to have one allele. And again, they're completely random. So gametes contain only one allele. So every time an organism reproduces, the chances of them having the exact same alleles in that gamete are extremely rare. It's completely random, okay? Now, the last thing that we're going to talk about, here's the vocab. So this whole section on the right, super, super important. Every organism that we study has two alleles. One from mom, which we know, and one from dad. Now, the vocab that I'm going to be using, you guys are going to have to be giving genotype and phenotype ratios for every example. So the genotype is the specific combination of alleles. What two alleles do they have? There are three possibilities for genotype. They're either going to be both capital, meaning both dominant, which we call homozygous dominant. So this is purely homozygous dominant, both capital letters. They could have one of each, which we call heterozygous. Grade 9 science, you guys might have called this a hybrid. Please don't call it that anymore. That's not what it is. It's heterozygous, hetero meaning a mixture or both, so it has one of each. Or it could be homozygous recessive. Homozygous recessive. Now, the other important vocab for you guys is phenotype. So the phenotype is different because it is the physical. So you see how pH in phenotype, pH in physical, that's something that can help you remember the physical expression of the genotype. So it's physically what you'd see. What does the genotype come across as? Now, generally with phenotypes, at least starting off with, there's going to be two different phenotypes, either a dominant phenotype or a recessive phenotype. Because the homozygous dominant and the heterozygous both have a dominant allele present, that's the phenotype that you're going to see. So having that dominant allele means that the dominant will always be expressed. So big R, and big R, little r will both give you the dominant phenotype. And that could be, in this case, maybe round, big R for round. And then if we had little r, that's the only time that you'll see a recessive phenotype. And in this case, maybe it's wrinkled. Okay, so the only time you can have a recessive phenotype being expressed is if both alleles are recessive, okay? Now, the very last thing that's going to be discussed on this page, the only thing that we haven't talked about, is just generations. And this isn't a huge idea, but you'll hear me talk about generations, especially the F1 generation. So it's important to just explain that for you guys. So we always start out with our parents. So maybe this parent is big R, and this parent is little r. Just for an example, doesn't have to be, it could be whatever. It could be both heterozygous, whatever. You have your parents. That's what we call the P generation. So the parental or the P generation, that's the very first one. That's whoever you're starting out with. When we cross these two together and mate them, they're going to get offspring. So here's your offspring. And in this case, let's say that he is big R, little r. The offspring of the parental generation we call the F1 generation. It's called the filial generation is actually what it's called, but don't worry about that. That just means essentially the first generation. So maybe we take this offspring from the first generation and maybe we cross it with another one of the offspring. So like inbreeding totally does happen in plants, whatever. Or maybe we take something that's completely different and we cross them together, okay? But anyways, we cross something from the first generation, we have more offspring, we would call it the F2 generation, okay? So we always start with the parental. When we take the parental generation, breed them together, we get the F1 generation. If we take two offspring from the first generation, mate them together, we get the second generation. And this can continue to the third generation, fourth generation, fifth generation, whatever, okay? But just so you know, especially this F1 generation is going to be used for vocab purposes a lot, okay?